Welcome. My name is Katie Summers, and I am a research microbiologist at the USDA in the Animal Biosciences and Biotechnology Laboratory in Beltsville, Maryland. Today I'd like to talk to you about some of the recent work going on in my laboratory about the cross-kingdom interactions in the porcine gut and its implications in health and performance. In my laboratory, our overall goal is to find alternative growth promotion mechanisms in pigs. And if you're wondering why that might be, it's because estimates show that the human population will exceed 9 billion by the year 2050. And if this is true, food production needs to increase 70% by that time. That is not a small feat. We need to find effective ways of growing bigger, healthier animals and crops in faster ways. But keeping in mind the idea of One Health, that all of our health is connected. Human health is affected by agricultural health and environmental health. So a good example of that would be diseases such as avian influenza or swine flu as well. Those diseases of animals can also go on to affect human health through causing epidemics or pandemics. Other items to keep in mind, and one very dear to our heart in my laboratory, is antimicrobial resistance. Animals in agriculture are one of the main receivers of antibiotics in feed, and this is a problem as we see antimicrobial resistance increase globally. Now, as people eat these food products, we are concerned that we will see an increase in antimicrobial resistance in humans as well. And keeping all of these important things in mind, we are targeting the gut of piglets as a research target in my laboratory, seeing how we can harness growth promotion through interactions with the microbes and the host cells that are there. Today I'd like to talk to you about the weaning transition in piglets. This is the most stressful period in a piglet's life, making it a critical target for intervention. Because if weaning does not go well for piglets, if they catch disease or if they aren't growing because of these big changes, that's a loss to farmers, and they never make up that loss of weight gain that is started at that time period. So what is weaning? If you're not familiar, this is the period where a piglet is taken from its mother, typically around 21 to 24 days of age in the industry. And this results in massive changes to the piglet and its environment. It is put from milk from its mother onto a solid feed. It is then also removed from the mother's antibodies that are found in its milk, which can lead to a predisposition to disease at this time period, because when those antibodies are removed, diseases can come in because the piglet's immune system is not fully developed. Other changes are a change in penning, change in what pigs are in there. Perhaps it's their piglets from their litter, but perhaps other piglets are mixed in, which is a common occurrence. So massive changes resulting in stress. This stress can lead to intestinal issues such as tight junctions being leaky and massive changes in the microbiome in the gut as well. So massive changes in a piglet's life, high stress, a big predisposition to disease. And so that is the potential for farmers and producers to lose money because, for example, in this picture seen on this slide, this piglet is suffering from a very common weaning-associated disease called scours, aka diarrhea. Now, in the past, antibiotics were provided in feed to these animals to treat or try to prevent some of these weaning-associated diseases. And the farmers and producers happily found that we could increase growth with low-level in-feed antibiotics by up to 30%. However, recent federal regulations have stated that we are no longer allowed to put that into piglet feed. In-feed antibiotics were traditionally utilized to prevent those diseases and promote weight gain. And if you're wondering why there is a federal ban against in-feed antibiotics currently, it's important to remember the rise of antimicrobial resistance. We are seeing massive increases in antimicrobial resistance in many agricultural species, and we're seeing more of these diseases becoming an issue in humans as well. So this is a global issue that needs a global approach, and this was a great step forward in preventing the misuse of antibiotics in feed in agricultural animals. Because we are no longer allowed to use those in-feed antibiotics, we need to find targeted ways to promote piglet health, growth, and disease resilience. And my laboratory really focuses on the microbiome, or the microbial members in the piglet, specifically in the gastrointestinal tract. And we approach this in many ways, including in vivo and in vitro models. If you're not familiar with the microbiome, it has broad effects across the host. Now, some people think that these microbes in the gut just affect the gut, but that is not true. It is able to change the central nervous system. It produces vitamins that are necessary to the animal's health. It promotes the development of the immune system and provides homeostasis to immune responses. If an animal is grown without a microbiome, it does not have a normal and functional immune system. It prevents pathogens from coming in. If that space is occupied by normal microbes that belong there, they work as a barrier against some pathogens from getting in. They can produce short-chain fatty acids, which are important in gut health, and they can protect the pig intestinal epithelium from injury. And these are just a few of the things that the microbiome can do. They have broad effects across the host. And so my laboratory likes to focus on that quite a bit, and we do separate it further than some groups do. We separate the bacterium, or the bacterial members, from the mycobiome, or the fungal members in the gut. And we look at temporal development and what is a normal microbiome in these piglets, and how is it communicating with those pigs, and how are these microbes talking to each other. Cross-kingdom interactions here in the gut have high potential for intervention. In vitro, we use a very traditional IPAC J2 porcine jejunal cell line. And we also do biofilm competition assays, and I'll talk to you a little bit more about some of these in addition to comparative genomics. You may be wondering why we separate the bacterium from the microbiome, and I'd like to tell you a little story about that. But to start, some things were known about the bacterium in piglets. We knew that it showed a reproducible succession pattern from initial colonization. We call the bacteria that get into the gut first in piglets when they are farrowed the founder species. And those founder species go on to create anaerobic conditions that permit the colonization of new bacteria in the gut. And so this is called bacterial succession, and it's reproducible. And we can see from farrow to growing and finishing stage a very reproducible bacteriome pattern. And some of the most abundant phyla are the Bacteroidetes, the Firmicutes, and the Proteobacteria. We knew factors like food source, geography, genetics, stress, and age could alter the bacteriome, but oftentimes it rebounded quite well. Now, when I started out at the USDA 5 years ago, we did a preliminary metabolome study that showed us some very interesting results of piglets during the weaning transition. When we assessed the metabolome of piglets throughout the weaning transition, looking for metabolites or markers that were associated with growth performance, both increased or decreased, we saw very few things, but what stood out to us was histamine. And we were surprised by that. So we went back to some pigs and we sampled their blood and we looked for histamine by ELISA. So a very simple approach to look at histamine levels versus growth rates. And so what we saw was when we separated piglets that were defined as slow growing versus normal performing piglets, elevated histamine levels at birth were associated with reduced piglet growth. So day 1 and day 21 are when we sampled the blood. And you can see here day 1, so the day of farrow, we sampled piglets, and on day 21 as they were being weaned. And so this was interesting to us because what is histamine doing? And we hypothesized at that point that the fungi in the environment might be colonizing these animals. And perhaps this is promoting mast cells to release the histamine. And perhaps this is an allergic disease, reducing animal performance, or what is called a failure to thrive. Failure to thrive is oftentimes associated with asymptomatic or low-level fungal infections that can be treated with an antifungal to then fix the disease, and then the animal or the human or the mouse, whatever your model is, goes on to catch back up with growth. So this was very interesting to us, but almost nothing was known about the microbiome in piglets, and so that is what we attempted to assess next. Sequencing the entire kingdom of fungi is not an easy task and has many potential complications. So while we worked on analyzing different primer sets and DNA extraction kits and databases for analyses, we began with a basic culture-based approach. And so we cultured fecal samples from piglets across the weaning transition. Here on this graph, you can see days post-farrow. So day 1 of farrow, across time, day 21, is marked with a red arrow showing the day of weaning, and then 2 weeks later, day 35 is where we ended the experiment. And so this is log CFU fungi per gram of feces. And what is interesting is that you can see from farrow to weaning, we have very low levels of colonization of fungi in the feces. We see some flux in that first week, and it's fascinating. I'll talk to you a little bit more about it later, but that composition is really in flux. This is a population trying to establish a community and battling out for turf in that gut. Then at day 21, we see a drastic, significant increase in fungal populations that actually stays at that level throughout the rest of the lifetime of that piglet. We've gone on to later studies in different genetics, and we see that this level is pretty standard across the board. So where is this fungi coming from? We sampled some of the items in the environment. It was not in the water. I don't have it on here, but we filtered air as well. We were not seeing that. The colostrum and the sow milk did not have large levels. And then piglet food stored and sow food stored, those did not contain any culturable fungi at all. However, when piglet food is placed into a pen, within 24 hours we see reproducible culturable fungi. So something is providing that. At this point, we presume things like the sow's feces contaminating the pen, piglets could get it from their mother, or perhaps the antibodies in the mother's milk are reducing the fungal burden, and then after the antibodies are taken away, the piglets are then able to be colonized. Or perhaps it's as simple as the fungi really enjoy the nursery feed that the piglets switched to post-weaning. So all of these were potential hypotheses of what was happening. At this point, we had our sequencing up and going, and we wanted to assess what was happening in the microbiome in these piglets to really establish what is a normal microbiome. So we sequenced with 16S primers for bacteria and ITS primers for fungi, and we assessed the different organ sites at day 35 in piglets to see what was normal post-weaning. And here we are assessing alpha diversity. This first graph is the Shannon index, and you can see as we go across from stomach to colon and then feces at the end, that each individual circle represents a different piglet, and the Shannon index for those piglets. So you can see for the purple bacteria that we see an increase in the Shannon index or alpha diversity for bacteria as we go longitudinally across the GI tract. However, we see an opposite trend almost for fungi, where the Shannon index is actually most high in the stomach of pigs. And that's really interesting to us. We know a lot of fungi have adaptations to permit them to grow in very acidic environments, and so perhaps this is coming to play, or perhaps they're just able to stay in that stomach location better than bacteria for unknown reasons at this point. We also looked at the observed ASVs, so amplicon sequence variants, and you can see the same sort of trend that we saw more observed amplicon sequence variants or types of bacteria over these organs versus the fungi that showed the highest level in the stomach. People always question perhaps these fungi are just transitioning through and none actually stick, and so we wanted to look at the organ variation of the microbiome. So here we have an NMDS plot looking at organ variation in these piglets as well. So the stomach is represented in the red color. The 3 different litters utilized for the study are shown in different shaped icons. You can see the upper GI tract, so the small intestine, the duodenum, jejunum, and ileum represented in green. The lower GI tract is in blue, representing the cecum and colon combined, and then purple represents the feces. And so here you can see that each of these organ locations has distinct populations of fungi present. This is not a total overlap where these fungi are just traveling through the gut, never establishing. They are finding niche environments within the gut that allow them to persist in these environments in a reproducible way. So this was an important point to make as we see that the fungi are not just transitory. We have also assessed the bacterium in temporal development. So here you can see Shannon index on the top, evenness on the bottom. These are the means of each of 3 litters. So we actually sampled the same piglets from farrow all the way to 2 weeks post-weaning. These were weaned at day 21. You can see the bacterium. There is a jump in the bacterium at the weaning transition, and we see that across the board for the 3 litters. When we look at the microbiome, we actually see the opposite trend. We see a reduction in alpha diversity at the point of weaning, and we also see a reduction in evenness. So these show very distinct patterns of colonization that are not equivalent at all. So when people consider the fungi to just be doing whatever the bacteria are doing, that's really not true, and there are more complicated interactions happening here. Here is our NMDS plot demonstrating the beta diversity, showing distinct patterns of development for both the bacterium and microbiome. So this is temporal, showing from day 1 to day 35. We have also added in adult sow and gilt fecal samples here in this bright blue, or feed samples in gray. Now, feed samples are somewhat difficult to get sequences out of that are of any quality, but we did see a little bit of reproducible fungal sequencing samples in the microbiome here in B. You can see 3 gray samples that are bunching over with the adult blue samples and the day 35 bright pinkish purple samples. So that was interesting to see. If we start over with the bacterium, you can see from day 1 in the bottom left, developing over time, distinct populations of bacteria are present in the fecal samples of these piglets. You'll notice a distinct separation between pre-weaning and post-weaning times here. So day 24 on, when the diet has changed dramatically, they are looking more like the adults, so very feed-driven. Now, the microbiome is not as clear, however, it also demonstrates the same pattern of development. So you can see a distinct pre-weaning, post-weaning group that is reproducible across litters. So this is 3 litters as well shown in this sample. So we wanted to look, we know that these bacteria and the microbiome are all in the gut together. We know they interact with the intestinal lining. We know they're producing short-chain fatty acids that interact with the gut. And we know that there are these cross-kingdom interactions where these microbes can speak to each other through quorum sensing molecules and other metabolites. And so we wanted to look at interactions. However, we needed to use bioinformatics heavily because almost all of the members in the microbiome, fungal and bacterial, are not able to be grown in the laboratory. And so this is a distinct disadvantage to this field, and so we relied on some inferred interactions that could be taken from the data. Our first attempt at this was a SPAR-CC correlation network. We ran this in R, and it shows us inferred interactions between the bacterium and the microbiome. Now this is at the genera level, so pretty far down. And what we're looking at here are different kingdoms represented by node color. So the green circles represent fungi, and the purple represent bacterial genera. Now the lines are either blue or red, showing positive for blue or negative inferred interactions. Now the node size is proportional to the mean-centered log ratio abundance. So one of the things that really drew our attention was how there were some fungi here that had strong interactions. So for example, number 4 here is Hyphopechia, which is a poorly understood fungi, and its interactions in the environment are not really known, but previously it was thought that this was a fungi that could not establish itself in the gut of animals and just was a contaminant in feed. But it looks like the story might be actually more complicated for this fungi. Number 6 down here in green was an interesting fungi called Kazakhstania sleuthia. And so this was a fungi that had been studied by one laboratory over in Spain for a little bit, and it was found to be very dominant in post-weaning pigs. But they hadn't gone much further with it, so relatively little is known. But you can see it's having interactions that are negative, or excuse me, positive predominantly in this sample. Here's a sparse correlation plot showing bacterial and fungal interactions in the lower GI tract. So this is a combination of the cecum and the colon of these post-weaning piglets. And this is a lot going on, but you can see the fungi here listed, and you can see the bacteria across the top listed. So we wanted to focus in on Kazakhstania since it seemed to be having the most inferred interactions with the strongest level of significance. And so when we looked across, we were seeing positive interactions with things like Alloprevotella, which is considered to be beneficial. We have a small negative interaction here with Lactospiraceae. And we have positive interactions with Lactobacillus and some more Prevotella and Subdilogranulum. So all of these are thought to be pretty beneficial bacteria, and so this was interesting to us. Can we harness this more? One of the other fungi we saw was Aspergillus, and here you can see negative interactions with quite a few of the bacteria across the board. So as we look across that top line with Aspergillus, we see negative inferred interactions with Alloprevotella, Lactospiraceae, Peptococcus, Prevotella. We see some positive interactions with Fibrobacter and some unknown bacteria that aren't known well, and Ruminococciceae. So this was interesting to us that Aspergillus, which is a potential pathogen, was having lots of negative inferred interactions. But Kazakhstania, which has been found in other pigs and not causing disease, was having some beneficial interactions, and we wanted to see if we could harness that. In this slide, you can see an overview of those positive and negative interactions for those two fungi, just pointing out that Kazakhstania really did have more positive interactions than negative, while Aspergillus, a potential pathogen, had many negative interactions with the same types of bacteria. So if you're wondering what Kazakhstania sleuthia is, don't worry, you're not alone. Not much is known about it, and most people have not heard of it. And there wasn't much in the literature, so we went on to characterize it in the laboratory, and it's a very interesting fungus. So it's part of the Kazakhstania tellurous complex, which is known to have many environmental samples, and it turns out to be related to Candida species, such as Candida albicans. I like to think of Kazakhstania as the cousin of Candida, which is interesting because Candida albicans is in the gut, just hanging out, not causing disease, of most adult humans. And it is a potential pathogen. It can go on to cause diseases like diaper rash or other issues in immunocompromised patients like thrush. However, overall, we're colonized with it and it's normal there. So could Kazakhstania sleuthia be the pig version of Candida? We wanted to learn more about it. We know now that it is the most dominant post-weaning fungus. So once a piglet is weaned, that is there. And that seems to be true across geography. It's true in South America, North America, Europe, and it remains across genetics. So the different genetic lines of pigs have been tested, and we do see Kazakhstania popping up for all of them so far. Here's a micrograph showing this budding yeast. You can see the little baby yeasts by the arrows, and you also might notice that it's a dimorphic fungus, meaning it can also produce hyphae. Here you can see it's pseudohyphae under the microscope. You can see examples of the colonies growing on different agarose media types on the top, so you can get different colony morphologies based on the food sources, which is pretty typical. And we know that these cells are high in nitrogen and amino acids, especially lysine, which could be important in piglet production and growth. So that was very interesting. Here you can see a recent paper from Science Direct talking about food samples that have been found in sourdough breads. And you might notice there's quite a few Kazakhstanias there. So Kazakhstania has been found in a lot of sourdough breads and also in kefir, a yogurt type drink if you're not familiar with it. And so while Kazakhstania slufiae is not here, a lot of its family members are there. And so this makes a very interesting environmental fungi that we wanted to learn more about. While we were assessing Kazakhstania slufiae, we wanted to look at some pathogenicity and its growth curves and to see if it was demonstrating antimicrobial resistance that we're seeing in other important fungi like Candida auris, which is a pathogen coming up in humans now. So when we assessed the antimicrobial resistance, we utilized 4 different classes of antifungals and 2 different antibiotics as negative controls. So cefoperazone and ampicillin are antibiotics, while here you can see echinocandins, azoles, polyenes, and pyrimidine analogs as the different antifungals that were tested. And you can see across the board that Kazakhstania was susceptible to these different drug classes and did not demonstrate any resistance at all at this point. And as expected, they were not susceptible to antibiotics that target bacteria. So that was great to see that it is not an antimicrobial resistant strain in these pig-derived strains. So then we went and looked at in vitro growth curves in many different ways, comparing Candida slufiae, Kazakhstania slufiae with Candida albicans CHN1, which is a human pathogen that was isolated at a hospital. So here you can see Kazakhstania slufiae and Candida albicans and how they grow over time, and this is OD 600. And you can see here that Candida always outperforms growth density compared to Kazakhstania slufiae, and this was across all different media broth types, temperatures, and shaking and non-shaking conditions. So this was very interesting to see that that pathogen always outgrew Kazakhstania in the laboratory. So perhaps it's some sort of growth defect or reduced pathogenicity. Just last year, we sequenced the genome for Kazakhstania slufiae, and we published the draft genome in Microbiology Resource Announcements last year. We're currently trying to work on some genome annotation, but that is a big project, and so we would like to look for any potential virulence factors or other things that might allow Kazakhstania to be a pathogen, or perhaps it is not an opportunistic pathogen at all and might be a true commensal in the gut of these pigs that has true beneficial effects. But at this point, we only knew beneficial effects from inferred sequence data, so we wanted to look at that in the laboratory. So when we look at the sequence data, here's an example of 3 litters showing the relative abundance of the different fungi present in the gut and feces. The important thing to note here is this bottom color, this teal, is Kazakhstania. So you can see, regardless of litter, born at different times, different pens, they all have dominant Kazakhstania levels. Here we're showing the temporal development of Kazakhstania, so it is relatively low but able to be somewhat found until we get to weaning, where it really has a pronounced jump and becomes over 40% of the mean relative abundance in these fecal samples for animals. So we're seeing in sequence data that this establishes really nicely in the feces and the organs of piglets. Next, we looked at the biofilmability of Kazakhstania slufiae, and we assessed this by OD readings of 595 nanometers. And we saw that Kazakhstania produces complex and dense biofilms reproducibly in vitro in the laboratory. And we wanted to assess the ability of different bacteria to alter this biofilm, basically looking to see if it supported our inferred interactions that were based on bioinformatics and sequence data. So we took bacterial strains that are pig-derived, and we took Lactobacillus acidophilus, Lactobacillus fermentum, and Enterococcus faecalis. And as I said, they are pig-derived. And that is important because strains of bacteria can be very different compared to other strains of the same species. So it's important to have it from a relevant animal species. So all pig-derived, these are readily available on ATCC. And we took and we grew those up to density. And then once they had hit that, we took those bacterial cultures and we filtered them to take just the supernatant and no live bacteria. So we took those supernatants and we added them to Kazakhstania slufiae biofilms. So here on this slide, all four graphs represent Kazakhstania slufiae biofilms in the presence or absence of Lactobacillus acidophilus. What you can see is Lactobacillus acidophilus is that second bar, and you can see it significantly increases the density and complexity of Kazakhstania slufiae biofilms, while Lactobacillus fermentum does not result in any statistical difference, and Enterococcus faecalis actually antagonizes the density of Kazakhstania slufiae and reduces it significantly. And this is 6 experimental repeats combined, so this is very reproducible in the laboratory. And this was very exciting to see, this cross-kingdom interaction altering biofilms of this fungi that has the potential to be a commensal or beneficial to the piglet. And this basically supported our inferred interactions, suggesting that our bioinformatics data was accurate and helpful in showing us what we should be looking at as we go forward in looking at animal growth. So just to go over this part of the talk briefly, we see antagonism and cooperation between fungi and bacteria in the piglet. And we see this through sequence data, and we see it in vitro in the laboratory. And we're doing current feeding trials now to assess that further in vivo. We were able to assess the microbiome in piglet and determine what is a normal microbiome. And the important thing to remember that even though it's considered a rare biosphere, these are numerically inferior to bacteria. There are very low levels, making sequencing sometimes difficult, but they still play a vital role in animal health. They do not show a bacterial succession pattern, and we see alpha diversity goes from high to low in piglets, not like bacteria. And what I didn't have time to show you today, but we have found that the microbiome is actually more manipulatable than the bacterium, suggesting it might be a great target for dietary interventions or drug manipulations to alter the microbiome to then alter what's happening in the gut, promoting health and growth in the pig without the need for antibiotics. And so the bacterium, these alterations can be transient, so this is an important target of research. For the fungi, the conclusions and implications, I kind of mentioned them on the last slide, but I want to point out it is important for pig health. These aren't just transient, passing through, doing nothing. They have distinct gut environmental niche effects. There are distinct populations in each gut organ that are having different interactions with the microbes and the intestine or epithelium of that gut. They are more malleable, so we can perhaps harness this much easier than the bacterium, which seems to bounce back from different dietary interventions quite quickly. And please remember, Kazakhstania slufiae is the most dominant post-weaning fungus. We see no evidence of its ability to become a pathogen at this point. We are working to look at virulence factors, if they're present or absent, but we do know it does not show antimicrobial resistance at this point. And that's important as we go forward because it would not be a viable feeding option if it demonstrated any resistance at all. In the last few minutes of my talk, I'd like to tell you a story about a new project we have going on that utilizes bioinformatics to look at growth effects in piglets. Now, we started by looking at enteroendocrine cells. So here's a picture of them here from a recent paper in 2017, if you weren't familiar with them. And enteroendocrine cells are found in the gut and the pancreas of animals. And what they do is they create and release GI hormones and peptides. And this is in response to different stimuli, and they can release these hormones and peptides into the bloodstream for systemic effect, but they can also send it to the enteric nervous system to activate the nervous response. And so these can have broad effects that result in different growth effects as well. And we have known for a while the microbiome has demonstrated relationship with feed efficiency. So we wanted to look at growth rate and feed efficiency, and if it's linked to interactions between the microbiome and those enteroendocrine cells through the enteroendocrine system. And so we're looking at things like short chain fatty acids, biogenic amines, neuroendocrine secretions, things similar to that. So very simple experimental design. This is with 48 piglets. We followed them from day 1 to day 35, typical day 21 weaning. Now on the days shown, we took weight, feces, and plasma from every single piglet. And the feces were then sequenced on an Illumina-MiSeq platform with 16S primers. Weight, a growth rate determination, was determined if they were poor performers, normal performers, or high performers for growth. And then plasma was used for ELISAs to assess hormones and peptides. So straightforward, but then we utilized the 16S data in CHIME and R to then look at different correlations. Here we took the 16S sequence data and looked at correlations between weight gain and the amplicon sequence variants. This is a Spearman rank correlation plot showing significant correlations that are FDR corrected and a p-value of less than 0.05 or more between change in percent relative abundance and percent relative weight gain between time points. So we grouped time points to be day 21 to day 24, day 24 to day 28, and day 28 to day 35. So the red circles indicate negative and blue circles indicate positive correlations, and the size of the circle indicates that strength of correlation. This is a neighbor joining tree with bootstrap values. We set the bootstrap to 100 and the values less than 50 are not shown at all. And this is based on the muscle alignment of the 16S rRNA-V4 gene region, and it depicts the phylogenetic relationship between the ASVs. So we have this tree and we have the correlations here. And what you'll notice is that Prevotalaceae NK3B31 had the greatest correlation with weight gain at early age. So the first column, as you look down, you can go down and see dark blue circles, and almost to the bottom is Prevotalaceae. And that blue circle is the greatest correlation at weight gain in that first group. And you can see Velanelaceae also had effects too. They were just a little bit less. And you can see these different correlations based on age. Obviously, as we wean at day 21, this early age group, day 21 to day 24, is in flux. We see those microbiome changes, and so this is probably not surprising that we're seeing lots of effects early on. You can also see at that next time point, day 24 to 28, we have lots of beneficial effects seen with things like Blautia or Megasphaera and Lactobacillus reuteri. And then at that last time point, we see some negative interactions with Ruminococcaceae and Chrysalinellaceae. And so we want to look more deeply in the future at the role of the diet change to the complex polysaccharides available in the gut, and maybe that is promoting the utilization of Prevotalaceae at that early time point. Here we looked at the effect of peptides and hormones on age, and you can see insulin-like growth factor 1, glucagon-like peptide 1, glucagon-like peptide 2, and glucose-dependent insulinotropic polypeptides. Here on this screen, you can see the groups labeled by day. So we had day 1 in red, day 21 in green, and day 35 in blue. The higher concentrations of GLP-1 and GLP-2 characterize the day 1 fecal communities here, while GIP levels had the strongest relationship primarily with samples ordinated with the day 21 cluster on this NMDS plot. So these are important peptides that we can look further into their role in growth in piglets during weaning. So we're utilizing this data to understand which microbes might be important based on bioinformatics, and we want to harness these interactions to promote disease resistance and growth promotion in a world absent of antibiotics in feed. So this slide will just tell you some of our overall research focus, like I've talked about, and some of the methods and aims we have to go forward. We're looking at the microbiology of the gut to determine those cross-kingdom interactions. We are looking at identifying alternatives to antibiotics through bioinformatics-based approaches through a machine learning approach. So we are taking large data sets, including but not limited to the microbiome, the metagenome, metatranscriptome, the proteome. We are utilizing machine learning models to see if we can get predictions of growth performance. And so we will use that to make targeted approaches going forward. We're looking at the effects of these candidate alternatives to antibiotics on the innate immune response at the gut mucosa. We can do that in vivo and we can do it in vitro using the IPEC-J2 line. We're looking further into Kazakhstania sleuthiae using bioinformatics. Again, we need to annotate the genome and look at the ability genetically of that fungi to promote health in pigs. We're looking at mechanisms of action of antibiotics. And we're also looking at different probiotic candidates that have been identified, such as Clostridium scindens in pre-weaning growth and health. And the fun part about working at the USDA is that they really promote data and science that is high risk. So looking for the mechanism behind antibiotic-induced growth is an exciting and very difficult project, and they give us the space and the resources to target big projects like that. And that's what's so exciting about being in a laboratory at the USDA right now. With that, I would like to thank and acknowledge the people who have helped me perform this research and analyses and my collaborators in funding. Former lab members include Dr. Ann Arfken, who is now at the CDC and who did a great job with a lot of the fungal analyses very early on, Julie Foster-Frey, a previous lab manager, and some of the students who helped with the data shown here today. My collaborators include people at the USDA and externally. I'd like to thank the Michigan Microbiome Project for their support in some of the sequencing startup projects and giving advice on ITS. And I'd like to thank the national programs for an AMR ATA grant that funded the large feeding trial in piglets. It's ongoing. And lastly, I wanted to mention that I have 2 current postdoctoral opportunities in my laboratory that I'm very excited about filling. I'm looking for an immunology-based postdoc, but I'm also looking for a bioinformatics scientist that might be interested in some machine learning and other bioinformatics-based models in piglets. So if you're interested at all, please don't hesitate to contact me. My email is shown here. It's katie.summers@usda.gov. And with that, I thank you for your time.